on the computer. Okay, so thank you so much, Dr. Yi, for coming here today. This is Angelman Updates, which is a joint production of the Angelman Syndrome Foundation and the Foundation for Angelman Syndrome Therapeutics. I am Dr. Jo Bichell. I'm a neuroscientist, but I'm also the parent of a son who's 22 years old with Angelman syndrome. And so I get to ask you questions a little bit from both perspective about what you're doing these days and what new research you're working on in the field of Angelman syndrome. So if you could just introduce yourself for a minute and tell us a little bit about yourself, and then I get to launch into the nosy questions.
Okay, sounds good. Like Terry Jo said, my name is Jason Yi. I am an assistant professor of neuroscience at the Washington University School of Medicine. I got into Angelman syndrome research as a postdoctoral fellow. Well, first, let me say I did my PhD with Mike Ehlers at Duke University, and then I did my postdoctoral training with Mark Zilka at the University of North Carolina at Chapel Hill. And it was during my postdoc when I really got into Angelman syndrome research by studying the UBE3A enzyme. So I've started my laboratory here in 2017 at Washington University, and we continue to work on understanding the regulatory mechanisms of UBE3A.
So right now, you are funded by the Angelman Syndrome Foundation, and we'd love to hear about your project that you're working on, but you were originally funded by the Foundation for Angelman Syndrome Therapeutics back in your postdoc days?
Yeah, so I was very fortunate to have the support of a lot of support from both foundations. So I was a postdoctoral fellow with the Foundation for Angelman Syndrome Therapeutics during my postdoc. They funded a lot of the research that I did in Dr. Mark Zilka's laboratory. And as an assistant professor, I've been funded by the Angelman Syndrome Foundation to continue our work into UBE3A.
That's fantastic. So tell me, what are you doing right now? What is the project that you're working on?
Yeah, so we're focusing on something known as variants. So these are small changes in the UBE3A gene. Typically, they will change one amino acid in UBE3A. The importance of studying this is that they're largely understudied, these types of mutations. So there are individuals who have Angelman syndrome who have these small changes in the UBE3A gene. Oftentimes, we don't know the significance of these small changes because they can occur in just normally between people. Sometimes they can cause disease because they can cause what we know as loss of function or gain of function of the enzyme. And so for a lot of people with these small mutations, they don't typically have a diagnosis because the genetic changes, it's not clear if they're really pathogenic or if they're just naturally occurring changes.
So in other words, you're not studying deletion positive cases, and you're not studying imprinting center mutation or UPD cases. You're studying UBE3A mutation, and you're not studying UBE3A deletion. You're studying the point mutations that just take out one protein. What happens if you take out just one protein? Does it change the shape of UBE3A? Does it change what it can stick to? What does it do?
Yeah, so let me just start by saying that this is a big problem as more and more sequencing is used in medicine. So we're starting to find millions of these variants across different individuals, and we don't really know what they are. So in the case of UBE3A, we already know that there's hundreds of these variants that have been reported in the literature, and some of these variants occur in people who are affected. So they can't really establish, though, because they're not deletions, like Terry Jo, like you said, what they're actually doing to the enzyme. So we take these mutations, we introduce them into UBE3A in our laboratory, and then we assess them functionally to see how they affect the activity of the UBE3A enzyme. And so we've been able to find that a large number of these mutations cause the enzyme to not function very well in terms of its ability to ubiquitinate substrates. They don't work very well. In about a quarter of the cases, the variants don't seem to do anything to the enzyme. And surprisingly, in about 12% of the cases that we can find, they actually cause the enzyme to work too well. So they cause a gain of function of the ubiquitin ligase activity of UBE3A.
So in those ones that cause it to work too well, do those patients who have that mutation, do they have the symptoms of Angelman syndrome?
Yeah, so it's been difficult to get that type of information, but we've been able to follow about 12 individuals with these types of individuals with these mutations. And let me just say, these are fairly rare cases that we see. So we're not going to get large aggregate data from a big population of patients. But as far as we can tell, they're all affected. The mutations pass from on the maternal allele. And none of the individuals that we found had a diagnosis of Angelman syndrome. And so they seem to have a different phenotype than Angelman syndrome.
Huh. So it's more mildly affected, those kids, or is it more like Dup 15? Because they would be patients that have an excess of UBE3A.
Yeah, we think it's more like Dup 15q syndrome. It's sort of this general, it's very heterogeneous. So let me say that. And in part, I think it has to do with how much UBE3A activity is actually being turned up in these mutations. So you could have very mild increases in UBE3A activity, or you could have very severe increases in UBE3A activity. So there's some heterogeneity just built in from that. But they tend to have diagnoses like intellectual disability to varying degrees. They tend to be epileptic, and they tend to have behavioral disorders. So you see a lot of autism spectrum disorder diagnoses popping up, some ADHD diagnoses popping up. So they don't seem to have sort of the classic phenotypes that you associate with Angelman syndrome, but they seem to have some other affectations.
So all these different point mutations that you're looking at, so some of them are what they call variations of unknown significance, I guess, right? The VUSs that everybody's talking about in the neurodevelopment world. And so we don't really know if they cause a real disorder or not. And in fact, probably some of them, the parents have the same variation of unknown significance, and maybe it doesn't cause a whole lot of symptoms. Is that right?
Yeah, so we were able to follow up until now 2 families where they've possessed these hyperactive mutations at least. And in each case, it was passed from the mother. So presumably the mother probably had it on the paternal allele and it was passed to children. Interestingly enough, though, in both families, kids who have the mutations are affected, whereas siblings who do not have the mutations are typically developing.
So it does matter. So what about the mutations that are not the that don't cause the UBE3A to work better, but all the other ones that you're looking at? Oh, or did I misunderstand? You're also looking at all the ones that cause it not to work as well, right? The point is there's some of them that cause it to work too well and some that okay.
Yeah.
So what about the ones that are not working quite as well? Some of them, are they all clearly causing symptoms, or some of them not so much?
Yeah, so it's hard to get that level of detail without actually knowing the physician who's seeing these patients. We sort of did that analysis in a different way. So there is a repository called ClinVar that sort of stores all of these variants that are being discovered. And they have the interpretation by the physician. So sometimes they're able to say whether they're pathogenic. Sometimes they can say whether they're benign. Usually it's, as you said, called the variant of uncertain significance, or VUS. And for UBE3A, about 2/3 of the variants in ClinVar have a VUS designation. So we sort of did the analysis by saying, okay, Angelman syndrome, the clinical manifestations are fairly recognizable. And so if there is a loss of function of maternal UB3A, a physician should be able to say, this child looks like he or she has Angelman syndrome. And so we sort of did that analysis. And for about half of the variants that we found to be loss of function variants, they're classified as pathogenic or likely pathogenic, right? And we think it's because the children who had those variants exhibited symptoms that were reminiscent of Angelman syndrome. For all of the variants that were gain of function, they were all VUS. So we think that that's sort of telling us that there is a very different thing that's going on in terms of loss of function and gain of function of UBE3A.
Wow. So it sounds like UBE3 is really picky. All you have to do is just cause one tiny little change and the thing is not going to work right, and it's going to cause a problem. But how does it cause that problem? Does it cause that problem because UBE3 can't stick to what it's supposed to stick to, or what is that little change doing?
Yeah, so for a lot of them, we don't really know. For some of them, we have some idea of what's going on. So one of the point mutants that we've been looking at, it's mutated to 3 different amino acids in 3 different individuals, right? So it's being mutated, and it's coming up as something is going wrong there. And as far as we can tell, it seems to affect its ability to form ubiquitin chains. So certain mutations prevent it from being able to build ubiquitin chains onto substrates. Some mutations cause it to do it too well. And so it's really good at doing its job. And so for that example,
Hold on, I'm going to stop you right there.
uh-huh.
So just to restate that a little bit. So one of the jobs of UBE3A, the Angelman protein, is to stick itself in a chain to a protein that is supposed to be destroyed by the cell. So it's basically, one of its jobs is to tag proteins for destruction, to show the cell what's trash. But
Right.
if it tags it just once, it doesn't consider it trash. In fact, if I recall, it actually makes the protein work better. Sometimes it turns on the protein. Once it sticks on 2, I don't know what that is, maybe some kind of gray zone. And then once it sticks on 3, then the thing is going to the dump, right?
Yeah, so typically, I think there are classic studies in yeast where they showed that it takes about 4 chains of ubiquitin on a substrate for it to go into the degradative pathway. And so building that chain becomes really important. And as far as we can tell, that's what UBE3A does. It builds these chains on the substrates and targets them for proteasomal degradation. And our data indicates that that's one of the processes that's really affected by these variants.
So what it's doing is it's either making the chain too fast, so it's throwing the wrong thing in the garbage, or it's not making the chain fast enough, so the trash is building up, right?
Right. Exactly.
Okay, well, so how come nobody can ever figure out what the trash is that's building up?
Yeah, that's a great question. And that's one of the long-term goals, I think, from this work, is that we can now create all these tools, these different forms of UB3A that do this work really well, that can't do this work, and that this will help us try to identify what's going on in terms of the substrates. But that's been a very difficult process to find out.
So how do you figure this out? Are you doing this on a computer? Are you doing this with cells that come from human beings, or how do you figure this out?
Yeah, so this is building off of work that I did when I was at UNC. So essentially, we use fireflies. We found the pathway that, at least in non-neuronal cells, where if you have a lot of UBE3 activity, it causes the production of a reporter enzyme. And that reporter, in this case, is the luciferase from fireflies. So it makes the cells glow. And depending on how bright the cells get, it tells us how well the UB3A is doing its job. So we are comparing to just wild type UB3A, and we can tell if it's not doing enough of its job. We produce dark cells. If it does a lot of its job, then it produces very bright cells. And so we've been screening different variants using that method.
Okay, so just to restate that, you took a protein from fireflies that glows and you stuck it onto UBE3A. And if the UBE3A tags something, or if the
We have the gene for the firefly luciferase on a reporter. So when UB3A is in the cell, it causes a transcription of this reporter gene. So
it's actually just if the UBA3A is there, if it hasn't destroyed itself, then you're going to have more of the glow. And if the UBA3A has somehow destroyed itself or has been destroyed because it's not shaped right and it's been thrown in the garbage, then you're going to have less of a glow. Is that right?
Yeah, so it's more to do with its activity levels. Because you can have lots of UB3A in there that doesn't work, and you won't have production of this reporter gene, so you won't have cells that are glowing. But if you have lots of UB3A activity, then it'll make more of this enzyme, and so you'll see more light.
So is that a robot that measures that, or is that a student that has to look under the microscope and go like, okay, yeah, glowing, okay, not glowing, okay, glowing? Is that how does it work?
So we do the assay, and we just do it in like a multi-well format in a plate, and somebody has to process that. But we're reading the light off of a plate reader. So it's automated in that sense. But before that point, we have to prepare the cells and process them to read that data out. And that's done by me and technicians and students.
So human beings, including you, get everything ready and then stick it in the machine. The machine measures how much it's glowing.
Exactly.
Okay, cool. So that's great. So it sounds like you could probably how do you get the way you get the mutations is through this giant database, which is ClinVar. So you don't actually need patients to come show up at the lab and donate blood.
No, we don't. Although there are a few mutations that we were able to get because patients were seen here at WashU. And so they're coming from different sources, not just strictly from that one database.
Okay, and so you're in St. Louis, and it's hotter than Hades there right now, if I recall. So plus I think you have an outbreak of the Delta variant of COVID. So what are you doing? What did you do for the 4th of July weekend?
Actually, we were driving back from Florida, which also has a Delta variant outbreak. We were going to see some family that we hadn't seen in a very long time. So we had driven down to Florida to see them.
So, and now what then in St. Louis, what do you do for fun? I actually grew up in St. Louis. I love
Oh, you
St.
did?
Oh yeah, I grew up I went to Kirkwood High School.
Ah, okay.
Yeah, and my three of my kids were born there, and St. Louis is a great town. You're right
Yeah,
up the
yeah.
road. Yeah. But so what do you do for fun? And I'll see if it's what I would have done for fun up there.
In St. Louis?
Mhm.
Um, you know, the parks are really great here. So we live near Forest Park. And so my daughter and I spend a lot of time biking around Forest Park. The cultural attractions are really great in the park, like the zoo and the museums, and they're all there's no admission to them, so we visit them regularly. Although we haven't in the past year very frequently. Yeah, and we live near what an area known as the Loop. So we within walking distance, so we do a lot of visiting restaurants at the Loop. Before the pandemic, there's also a concert hall on the Loop that I would frequent. But yeah, it's a pretty, I think, a pretty easy place to live.
It is. It's a great place to live. And the zoo is the best. When my kids were little, we would, I kid you not, go there 3 nights a week and have dinner there because it's free. And the
Yeah,
kids could walk around and look at the animals. And we went to the art museum once a week or something like that. We were always in that park. It's beautiful. So I think people don't realize that about St. Louis. Back in the day, it was like one of the world-renowned zoos. I don't know if it still is, but it's a gorgeous place. So
I think it's, yeah, it's also, it's, I've always been really impressed by the exhibits there.
so is there anything else you want to tell the community that I should have asked you that I didn't ask?
Um, hmm. I think that pretty much covers it. We're not working on anything directly translational. Although I think what we do may be important in terms of the clinical trials that are going on. I think sort of their success depends on recruiting the right people. And so you certainly don't want to recruit somebody who has one of these hyperactivating variants into these clinical trials. So I think there's some utility there for our work to sort of figure that out.
Wow, that's a great point. So, but not only that, I mean, I think the other main point is that sometimes basic science that doesn't seem like it's going to lead to a treatment
Mm,
is actually exactly what leads to a treatment.
yeah.
And so maybe one of these days, maybe you'll find a way to make the mutation on UB3 that makes it work better for people so that they can boost the activity of the UB3 they do have or something like that. And people have to sometimes be patient that even if it's not obvious, it might end up being the missing clue to the whole puzzle.
Yeah, I mean, I still remember uh, Xian Sung, who was a postdoc in Ben Philpott's lab, when he would come over to he used to come to our lab meetings at Duke in the Ehlers lab and pitching this drug discovery project and thinking that's never going to work. And it just, that completely changed the way we think about Angelman syndrome and the possibilities for reinstating the paternal allele. So yeah, you're right. You just never know.
And that was all about glowing dishes too. That if they hadn't done that particular experiment with the exact same type of cell prep and the exact same type of robot reading the glows in the dish, we wouldn't have
That's
right
right.
now thought that we could actually activate the paternal UB3A. So
Yeah,
yeah, you never know
never know.
what's gonna happen. So thank you so much for participating in the Angelman updates and for all the work that you do. And I'd love to touch base with you again in like 6 months and find out what's new.
Yeah, absolutely. This was fun. Thank you so much, Terry Jo.
Thank you. Have a great day.
Okay, bye-bye.
Okay, bye.